Homo neanderthalis, or Neanderthals as they are more often known, are an extinct species of human that was widely distributed in Ice Age Europe and Western Asia between 250,000 and 28,000 years ago. They were characterized as having a receding forehead and prominent brow ridges. In 1856 the first Neanderthal fossil was discovered in the Neander Valley near Düsseldorf in Germany. Since then, researchers have been striving to uncover the position of Homo neanderthalis in modern human evolution. Homo neanderthalis appeared in Europe about 250,000 years ago and spread into the Near East and Central Asia. They disappeared from the fossil record about 28,000 years ago. Their disappearance has been put down to competition from modern humans, who expanded out of Africa at least 125,000 years ago, suggesting that there would have been a period of coexistence. The questions arising from this coexistence is, did the two species interbreed? Have Neanderthal genes therefore contributed to the modern human genome? Well, you will find the answers shortly. Initial studies of DNA from the mitochondria of Neanderthals showed that their mitochondrial DNA looks quite different to that of modern humans, suggesting that Homo neanderthalis and Homo sapiens did not interbreed. In 2010, scientists from Germany and the USA sequenced the DNA of an entire Neanderthal genome. They also identified another archaic human group called Denisovans, named after the Siberian cave in which the fossil finger, from which the DNA was obtained, was discovered. In 2013 they obtained a more refined Neanderthal genome sequence from a 50,000-year-old Neanderthal toe bone, found in the same cave in southern Siberia. DNA can survive in bone, long after an animal dies. Over time the DNA from various microbes that encounter the skeleton will also invade the bone. As a result, the DNA can be contaminated with microbe DNA. Scientists therefore have to ensure that they sequence only the Neanderthal genome and get rid of any DNA material left behind by these microbes or resulting from contamination by modern humans who handle these bones. The genome sequence suggested that early modern non-African humans interbred with their now extinct ancient human cousins as they journeyed along coastlines and over mountains. Analysis of the Neanderthal genome revealed that the toe bone came from a woman as it had two X chromosomes. Further analysis showed that each pair of chromosomes was similar in sequence. This suggests that her parents were closely related, perhaps an uncle and a niece. Inbreeding is generally bad for the genetic fitness of a species as it reduces the variation in a population, making it more susceptible to disease and illness. This reduced genetic variation could explain why Neanderthals became extinct. When comparing human genomes to the Neanderthal genome, human genomes resemble each other more than any of them resemble the Neanderthal genome. Some Neanderthal DNA is similar to DNA from people of European and Asian origin, but these similarities are not seen in African DNA. This suggests that modern humans evolved in Africa and then expanded out into Asia and Europe, where Neanderthals lived. A degree of interbreeding between Neanderthals and early Homo sapiens then occurred in these areas. A study carried out in 2012 estimated that this interbreeding probably took place about 37,000 to 85,000 years ago, and it is estimated that the proportion of Neanderthal-derived DNA in people outside Africa is 1.5 to 2.1%. Nowadays, many of us carry a small fraction of DNA from our archaic Neanderthal and Denisovan ancestors. This shared DNA could have shaped our individual susceptibility to modern-day diseases or adaptation to new environments and climates. Scientists have found 9 Neanderthal genes in living humans known to be associated with susceptibility to conditions such as type 2 diabetes, lupus, and Crohn's disease. It has also been shown that high-altitude adaptation in Tibetans may be a consequence of archaic Denisovan DNA sequence in a region of DNA associated with hemoglobin concentration at high altitudes. Additional research is being carried out to investigate these links further.